Mostly the standard announcements. I think I've got all of your muddiest points back to you. I think the only grades out are there's 6 or 7 mini project sixes that the grader is working through grading. Otherwise, I think everything else is back. I finished the final exam, so it's posted, but not it won't be available until Monday of finals week, and then it'll be available all week. So there will be even though there is a session booked for this room, there won't be any in-person version for that. They'll all be at home. It's not proctored. It's just open book, open notes. It is sort of honor code, assuming that you'll work on it individually. It's meant to be an individual exam. There is a very similar exam that I've made as a practice exam as well that I released, I think, yesterday. So if you go to the final exam module, you'll find a practice exam inside there. You'll find 2 things. You'll find a practice assignment, which randomly draws from the previous concept checks. And so it's just an opportunity to sort of, get practice from those. And then there's a final exam, which is modeled after a final exam I gave, I think, back in 2022. And so that is going to be a very similar length and content as the exam that I've put together for this semester, which will be released on Monday, available through Friday night of finals week. It's a timed exam, so once you start it, you'll get 110 minutes. And then, so just make sure that your 110 minutes finishes before the availability window finishes, and then you won't get your scores until after the availability window closes. And that's pretty much it. So otherwise, there's these concept checks and mini projects that aren't due until basically the last regulation weekend before finals week. And course evaluations are open, so you should have gotten emails about that. It's also linked in the nav bar from the side course on Canvas. It says like home and then the course evals, and then, like I said, there's this practice final available. So any questions? Oh, regarding final projects, so they're due Wednesday of next week. And so I originally was planning on collecting them through Perusal. I wanted to try their peer review out, but I may decide that this semester, given how small the course is, that I'm just going to keep doing it through Canvas. So either way, on Wednesday, there will be an assignment, and it's probably just going to be upload to Canvas, where you're going to upload your presentation, these will be group uploads. So you'll upload your presentation, you upload your 4-page report, and then Wednesday night, then I will, once we get all those uploaded, then I'll go through and assign peer reviews. And so I would like to have every individual review 2 reports and 2 presentations, but that may, due to a technical limitation in Canvas, I can only auto assign when it's a group 1. So if I have the stamina to manually do the second, then I will, but otherwise I'll just do 1, and I'll let everybody know what the plan is. So at a minimum, at a maximum, you review 2, at a minimum, you review 1, where basically you're going to get an assignment where it's just going to ask you to review another report from another group and another presentation from another group, and there'll be a rubric, and you fill out the rubric and maybe add a few constructive comments. And then if I see that you've done that, filled out the rubric, added comments, then I'll give you the point in for you scoring your final project points for the peer review. And then when I get all of the review scores in, then I'll look at how all the other review scores were, and then I'll also review things myself, and then collectively come up with a score for the projects based mostly on me, but in part on the peer reviews, and then also the presentations, similar way. And so that's how you'll get your final project scores. So that'll go through me, not the grader, but I mean, there's peer review modulates that. Okay, so I think that's all of the policies and procedures. So any question about that? Okay. Feel like it's a little darker up here. I thought I hit all the lights. Let me try. Okay. Okay, so this is the second to last section in our neural computation and learning. So we've went through reinforcement learning. Now we're pausing here at supervised or self-supervised and unsupervised before we finally close with spiking and neuromorphic systems. So the big thing here is this discovering structure without a teacher. And so unsupervised learning and self-supervised learning are learning without labels. And so far, the types of learning we've been doing have been either supervised learning, so where we've got these external, I'm going to call them targets, that feed into loss functions, which guide the plasticity of the neural network, which update the weights of that neural network. And that includes temporal supervision. So it could be you're looking at an image and you're looking for patterns, or that could be you're looking at images over time and you're looking for patterns over time. But you have been provided guidance for for these stimuli, you want these outputs. And so as a consequence, then you gradually learn those associations. So we draw connections between supervised learning and classical conditioning, in that you're forming an association between a new stimulus. You see maybe this one, light. That's maybe better. Between classical conditioning, where you're forming an association between a new stimulus and an existing possible response. And so your classifier has 10 labels that it can produce. And without training, if you give it any input, it'll randomly produce one of those labels. But then through this loss function, then you propagate air backwards and inferences forward and new air backwards and so on and so forth through back propagation that washes over the neural network. And then gradually you make an association between the desired stimulus, or rather the stimulus for the desired response. So you say, no, these stimuli should go with this response that you already have in your vocabulary. These stimuli should go with this response that you already have in your vocabulary. So that's what we kind of associate them, so to speak, with classical conditioning. And we also have learned, of course, about reinforcement learning, where instead of having a target, you have external rewards. And so as the agent moves around and it does certain actions at certain states, then it receives rewards for those actions at those states, or potentially punishment, but it receives feedback from the world about the actions that it is taking. And so we liken that to operant conditioning, where we're updating the frequency of certain state action pairs in sort of which we would say kind of psychology or biology literature, certain stimulus response pairs. So we're saying that the animal normally doesn't respond this particular way to the stimulus, or doesn't usually have this response to the stimulus. This response might not even be in its vocabulary, but if we can get it to randomly do that response, then we're going to reward it. And then gradually it's going to start doing that response more and more and more. So that's kind of the difference between those two. But either way, in both cases, you have sort of an external target or pattern that you're trying to then freeze into the network, or you're providing external rewards and trying to get the network to change its behaviors based on those rewards. But both of these require effectively a teacher, but brains and then machines often figure things out without these external signals or guides. So how do you do this without a teacher? And why might that be useful? Well, if you think about it, so so unsupervised learning is sort of foundational in that if you think about a developing organism, they may not have any external cues initially. In order for them to even figure out how to find rewards, how to get those external cues, they need to start doing pattern learning. And that's kind of where we get unsupervised learning. So we're finding and representing hidden patterns that are in the world around without any guidance, because you may not even know how to receive that guidance yet. That itself might be something that has to develop first. So it's foundational. So you can build all of the other learning types on top of unsupervised learning if you kind of craft an architecture around them. So the goal for unsupervised learning is to group, compress, and discover structure from input. So you get inputs, but you don't get any rewards. And the common example to start with is clustering, or what you might call automatic classification. So it's like classification without labels. So what that might look like is you have an input that is, say, got this feature space, that these three-dimensional feature space. So you've got this feature, that feature, and another feature, and then points are scattered all around this feature space. And when you look at that in this feature space, you start noticing kind of clumps. And so you might see a clump that these in the feature space tend to be kind of, maybe I'll do a little different style here. So maybe these in the feature space are clumped together. These in the feature space are clumped together. These and these. And so you weren't provided any labels, but because there's all of this gap, then it sort of becomes natural to start grouping those together and generalizing and saying, well, we're just going to call that, we're mapping that to 1 in our heads, these to 2, these to 3, and these to 4. So we could have provide our own labels, our own classes that we weren't provided ahead of time. We don't even know how many classes we were supposed to get. We just sort of look at that and say, it looks like there's roughly 4 classes. And then from there, we can start building additional learning rules on top of that. So rather than, we can start to generalize. So if we happen to notice that that point 1 in our input is very close to point 6 or something like that, if we learned something about point 1, we might apply that learning to point 6 because they're now in the same class. So it makes other types of learning more generalizable later. And so, examples of this is like animal calls. So if you're listening to audio, so this is sort of related to temporal supervision of a forest, and you hear birds, you hear frogs, you hear crickets. There's a lot of things that you hear. And one question would be, how many species do I hear? And you may not know that number ahead of time. So if you just listen to that, then a common thing you can do is this clustering. So in an unsupervised way, you can then count how many things are out there. And you can say, my best guess is there are 10 unique species that are out there. You might be able to go farther in that if there's enough individuality within a species, so certain calls, like, that mockingbirds around here, I can hear them. I immediately know a mockingbird at the species level when I hear it on campus. But every mockingbird, because they're a mimic, sounds a little different. So at night, not when I'm if I'm up late writing a lecture or something like that, and my sitting in my living room, their male mockingbirds will often call all night. They're sort of sometimes known as the northern nightingale, and I know it's not just a mockingbird, it's that mockingbird. It's the same one night after night after night that is up from midnight to 4 a.m. with that same call. So if there's enough individuality, I can not only count how many species are out there, but how many individuals are out there. So so that's kind of another thing that I can do just with unsupervised clustering. I just start noticing, like, I've heard that before. And of course I didn't hear exactly that, but I heard something that was close enough to that my head starts grouping them together. The other one, a simpler example that we often use, a lot of people in this class are using this data set, IMNIS, so handwritten digits. So if you have a bunch of digits that are, it's a 9, but it's a 9 written all sorts of different ways, and you suddenly realize, wait, all of those things look similar. So we're going to group those into a 9. So, one way to do that would be with supervised. You can label them and say, all these are 9s. So learn what a 9 looks like. Or you could say, here's a bunch of digits. And then you can have it automatically say, in MNIST, all these up here, all these happen to be clustered together. I don't know what number that is, but it happens to be that all of these happen to be the same number. So then later I can see, oh, you've grouped all the 4s together. We're going to call that 4. Oh, you've grouped all the 3s together. We're going to call that 3, and so on. So that's kind of another example of that. Lots of different ways to do this. It's good old-fashioned machine learning stuff. K-means clustering, hierarchical clustering. So a lot of clustering algorithms are out there. But the point I'm just trying to drive home is no labels, completely unsupervised. And as we're going to learn, that there are plasticity mechanisms inside the brain that are very good at this. They're not doing k-means, they're not doing hierarchical clustering, they're doing Hebbian learning, which we'll talk about a little bit today, and then we'll focus a lot more on in our closing lecture for this unit on Thursday. Okay, so a related example, anomaly detection. So similar plot, I've got this plot over here where I've now got this feature space 1, 2, and 3 dimensions. And in this feature space, I've got a group of normal data that, I don't know, might be clumped close to a particular region of the feature space. And then I get some data that's way out over here. And so if I can manage to cluster these, then I might be able to not only cluster them, but actually say, wait, that cluster is the weird cluster. Those are anomalies. I've never seen that before, but it is not like the things that I expect. And that's anomaly detection. And so you might have training data where you say these are all normal. And then if you get anything that's far enough away from your training data, then you tag that as anomaly. And you don't have training data for anomalies. They're anomalies. They're just untrainable. There's just so many weird anomalies out there. It's not a class that you could train in supervised learning. So you just have to say, is it different than normal? So you either have to figure out what normal is by watching the operation over time and then build up a model inside your head of what normal is, or you might be given labels for normal data and then have to figure out what's abnormal. So what non-neural algorithm have we looked at already that does anomaly detection much earlier in the course? It's much more it's immune, or I would say it's genome adjacent, but it is not a GA. So it's an algorithm that we're saying a classic example was there's a drill press and and after listening to the drill press, then you eventually figured out the drill press was malfunctioning. And so this algorithm was inspired by something in vertebrate physiology that allows it to detect when things like drill presses and manufacturing systems and cyber systems have got anomalous behavior. Yeah. Negative selection, that's what I'm looking for. Yep. So that is a non-neural way to do that. So in negative selection, that is also viewed as another unsupervised learning approach where you might have a bunch of examples of normal behavior, self, and then you generate a bunch of classifiers for abnormal behavior. Get rid of the normal behavior ones, leave the abnormal behavior ones in, and if the abnormal ones hit, then that must be a weird outlier. And you're going to have a bunch of abnormal classifiers floating around that will never hit. But as they're constantly being generated, if they do match, then that's probably a good indication that you've got anomalous behavior. So that's one way to do anomaly detection, but you can also do it through kind of things that are clustering adjacent. So just a connection back to there. All right, any questions about clustering before we build on clustering? And usually probably pretty standard data science-y review stuff. Okay, so clusters. So how do we learn cluster without external labels is already hinted at. Hebbian learning provides us a way to do that. We're going to go into that in a lot more detail on Thursday. But the basic gist is that neurons that fire together wire together. And so we're going to introduce rules, both kind of simple rules, kind of like what you'd see in a recurrent neural network, as well as more complex rules like you'd see in a spiking neural network that implement this neurons that fire together wire together. And the idea here is that this neuroplasticity mechanism naturally clusters similar inputs together. In examples that we'll talk about on Thursday, you can basically shine an MNIST bitmap at a spiking neural network, and if it happens to see the same, we'll call it the same pixels light up over and over again, it will start to associate the pixels that light up together, like, oh, that's a 9. The pixels that happen to light up with a 9 will tend to end up getting mapped without any supervision to a particular output. And then other pixels that light up when it's an 8 will get mapped to another output, and so on. And this just comes from if you've got a neuron tied to every pixel, so just black and white here, then if a bunch of pixels tend to fire together because that's just part of the letter, the 9 just happens to excite these pixels, then over time memory traces will get reinforced by this Hebbian learning, by this neurons that fire together wire together, and then that's going to end up creating an association with a kind of an internal latent grouping, which then will cause the outputs to have a discrete pattern relative to that group of inputs. And then you can learn that pattern. And you may not know, is it a 9, is it an 8, or is it 7? You might have to figure out what it is. But you do find that you can just, with this simple system, you can learn these things. So people have built hardware using spiking neural networks spiking neural networks implemented with these things called memristors, which are just kind of like they're called memory resistors. They're sort of a combination of a resistor and an inductor. And so they're just plain old hardware. And you shine these bitmaps on them, and then over time they learn, I can learn MNIST. And so without any computational, no iterations, no update rules or whatever, then this just chips on a breadboard, then gradually learns how to do MNIST in an asynchronous way, which I think is pretty cool. But we'll talk about next time. So, but the point I'm just trying to get at is that this very simple underlying mechanism that automatically updates the synapses, the synaptic weights in your neurons, just as a natural clusterer. It's just very good at things that happen at the same time, forming memory traces that end up becoming the latent representations of these clusters. And that's, again, what we'll talk about on Thursday. But brain architecture can make this easier. So it's one thing to just say we've got just a dish of neurons and we're going to shine light at it and hopefully it figures things out. But if you organize those neurons in particular ways, then you can make it so that you're more likely to learn certain things using these mechanisms than others. And so that's what I want to talk about as we're moving sort of into more advanced unsupervised learning. And so dimensionality reduction. So is the next kind of step here. So I sort of skipped over, again, we'll talk about this more on Thursday, but before I step away, if there are any sort of, basic questions about this idea or clarifications, does anybody want me to bring up any questions before I move on to, all right. So very simple mechanism. How do we make it a little more useful? We can build other things on top of it. And one example of that is dimensionality reduction. So another example, unsupervised learning, again, a lot of this will be review, and I apologize for that. I'm just trying to build it into a cohesive message that I can link back to biology and eventually hopefully build up to something that's new. So let's say we have here a 4-dimensional space, 4 dimensions because it's 3 plus color. So that's why I've got P for purple. I've got these numbers. So 1p, that's just supposed to be 0.1 that happens to be purple, 1p, and that it is colored purple. 6 and 7 are blue, 10 is green, 4 is green, 5 and 8 are orange, 9 is red, and then 2 and 3 are purple. So 4-dimensional space here. So 4-dimensional data. I look at that, and already I'm kind of doing dimensionality reduction because I've depicted 3D, I've depicted 4D on a 2-dimensional space. And so it's natural to look at that and see clusters because I've squished them onto the plane. But if you actually were to look at that out here in 3D, so if in some classroom of the future, then this thing was rotating out in front of me, and I kind of could move it around and things like that, then it might be harder to see those clusters. So what we do then is this multidimensional scaling for visualization. So there are different forms of dimensionality reduction. And when we're trying to visualize clusters, then we use tools from MDS, multidimensional scaling. And the idea here is I want to take these 4-dimensional entities and map them to a 2-dimensional space. And I want to do that in such a way that I try to make the clusters easier to see. And I try to do that so that hopefully, even though the positions have to change, like in order to go from this 4-dimensional volume to this 2-dimensional plane, some distances are going to have to change. But hopefully, I can organize them in 2D so that their pairwise 2D distances are very similar to their pairwise 4D distances. And if I can do that, if I can say that these pairs are closer together, then maybe I can more obviously see clusters in this space. So maybe now I can go into here and say, well, I wasn't quite sure in 4D, but in 2D, then I'd say this is a cluster, this is a cluster, this is a cluster, this is a cluster, and this is a cluster. So this input transformation made clustering easier. So going from high dimensions to low dimensions makes it easier. Turns out it also gets rid of a little bit of noise. So there could be variation here that after I go through this remapping, then that variation gets washed out in the attempt to try to get these things as close to the distances as possible. So it hopefully maintains the important distances, I guess, differences, while washing out some of the less important ones. And so a good multidimensional scaling tries to maintain between object distances approximately. So that's sort of the idea here. And there's a bunch of different ways we can do that, and that's what we're going to talk about here next. But that's the point, is hopefully this point is clear, is that if you can take things from an ugly, 1,000-dimensional space and project them down to 2-dimensional space, at least visually, it's a lot easier to see potential clusters. And arguably, even for a machine, if you do this dimensionality reduction, it will be a much more robust clustering engine than it would be if it tried to cluster on this space here. So questions about that perspective? Okay. So like I said, a good MDS between object distances are approximately preserved. The simplest way to do this, not very neural at all, just good old-fashioned stats, is to leverage the principal component analysis. So the idea there is that you design, again, you've taken basic stats course. This is all old, information, but just the level set. Design a linear transformation T, so that we've got two inputs here, X and Y, in my original high dimensional space. And I'm going to transform them into a new space, which is going to actually be the same number of dimensions. And so I just do a linear transformation on both of those. And I'm going to design that linear transformation so that certain things hold. One of the things that's really important is the angle and length is preserved. Or saying that another way, the inner product of a vector on another vector in the original space is equal to the inner product of the transformed vector in the new space. The transformed vectors in the new space. So it doesn't actually, it might rotate things around, but it's not going to squeeze anything. It's not going to stretch anything out. It's basically just going to reorient the space. That's basically all this is saying here. Angle and length are preserved on everything there. But what does it do? But in that space, we've reorganized the dimensions. We've reorganized the coordinate frame. So really, we're not moving the data. We're just moving the coordinate frame. So we used to have whatever X, Y, and Z, but now we're going to have principal component 1, principal component 2, and principal component 3. So we still have a 3-dimensional space, but we've now just rotated the space to reorient it along the dimensions of the data. And what I mean by dimensions of the data are the dimensions of greatest variance. And so and I'll draw, maybe there's a little diagram will help. So if I have a data set here, this is my original data set. Well, this is the shape of my data set. And who knows what it looked like in the original space, but I mean, it looked like this, but it was just rotated around. But now what the PCA has done is it's taken this bread loaf, this football, this rugby ball, whatever you might view it to be, and it's moved it so that the longest, the PC1 corresponds to the most variance in the data. PC2 corresponds to the next most variance, and PC3, you can't hardly even see it, corresponds to the least amount of variance. And so by doing that, and I think I've just got those written here, most variance PC1, least variance PC3, and the intermediate variance PC2. And I can even say how much variance is explained by this principal component. And I can say that 70%, 100% variance. So if I was to say, like, all of the variance in the data, if I account for PC1, then the remaining variance left over after I account for PC1 is only 30%. So that means that 70% of the variance is controlled for, is accounted for by PC1. And then of the remaining 30, then most of it goes into PC2, and there's a very little amount left for PC3. That's what a PCA is doing. So with that, then I can take that PCA and I can say, well, in this particular case, this third dimension, there's really not a lot left in there. So how terrible would it be for me to just forget that PC3 exists and just project everything on to PC1 and PC2? Kind of like when we were looking at that four-dimensional example on the previous 2 slides ago over here, and I was saying it already looked like clusters were obvious because our brains were projecting it onto the screen. Well, what if we've sort of reorganized that so that it was okay to do that? That's what this basically does, is that when the higher principal components have very little variance, you just get rid of them. And then you have a simple dimensionality reduction where then you assume that only these two components matter, and then try to do whatever analysis you want on what's remaining. So, and so that's our MDS approach. Discard the upper PCs, and here discarding PC3 does not affect the shape very much. And so, I meant to highlight that earlier, but yeah, so we preserve variance, but once you discard, you do deform things a bit because there was important variance in PC3, but it was negligible. So we're actually picking up noise by discarding this PC3, but we're not picking up that much noise by getting rid of this information. And so, but then maybe the ability for us to work in this space is counterbalances the extra noise we've added by getting rid of PC3. That is one relatively simple way to do multidimensional scaling, still very popular, particularly in the sciences. Not quite as popular in computer science because there are better ways that are more algorithmic, and that's what we'll talk about next. But any questions about the PCA? So it's a major tool in stats. All right, so what's a better way to do it? So t-SNE is t-distributed stochastic neighbor environment. I don't know, I'd have to, but t-SNE is basically somebody said, let's do this in a more responsible way. Rather than using a tool meant to do one thing and then, kind of manipulating it to do another, let's just build this is, I think Hinton actually did this. Let's just build a tool that is meant to do exactly what we said. You want to produce two dimensions, and you want you to preserve the distances as much as possible. We'll just write a little optimizer to do exactly that, and that's basically what t-SNE does. And so this is not the simple linear transformation of the PCA, so it does not maintain variance. But it does a pretty good job for visualizing data at doing exactly what you want it to do. So it's just, I don't want to go into great detail here, but it's just an approach specifically designed for dimensionality reduction to maintain distances. So give me the best two-dimensional representation of these data that maintains those distances. And there are other approaches as well that, UMAP and other things like that do something very similar to this, but t-SNE is kind of a go-to. But when you look at a plot from t-SNE, it often looks like the same thing people do for PCA. Practically speaking, for a lot of data sets, you're not going to see things that look that different or that functionally different from whether they're t-SNE or PCA or others. But theoretically speaking, t-SNE is better for visualization. But let's say, again, I was talking about that's for visualizing, but I am thinking about what are architectures, neural architectures that can help with clustering, actually doing the decision making, downstream in your head, not, for just visualizing data. And that gives me the opportunity to introduce the autoencoder. So If we want to pre-process data for clustering, then what we would ideally like is a transformation into some lower dimensional latent space, unobservable space, not used for visualization, that compresses the data, gets rid of noise that is not helpful for clustering, and amplifies the things that are similar among those entities, so that you can have these robust clusters later. And that's what kind of the autoencoder is meant to do. So it in part captures shared patterns, but also in part is a filter, and then it gets rid of irrelevant stuff that doesn't help you with just the discrimination. So what does an autoencoder look like? Well, and again, this might also be review, but the idea is you have an input, high dimensional input, goes into a multi-layer perceptron, just a feed-forward neural network. We're still going to call this unsupervised learning, though, because although we're going to train this network with backpropagation, we're going to do it in a way that does not require us to have any label information ahead of time. So we build what's first called an encoder, which is just a multi-layer perceptron that gets smaller and smaller as you go inwards until you land the output of the encoder over here. This little Z that I'm calling it here is a low dimensional representation of the input. Now, our goal is to build this encoder so that this low dimensional representation captures all of the relevant features of our input and throws out the kind of stuff that's not very informative. Well, how are we going to build that? Well, encoder suggests that if we just had a decoder, we could then look at what our internal representation produces if we reconstruct the input and then compare the two. And that's what we're going to add. We're basically going to create a bow tie here where the output is a mirror of the input. And we're going to train this network so that we're going to try to bring the input and output of this encoder decoder structure together. And so the way this looks like is we just measure the input and output, and we generate a difference, which we then can run into a loss function that we can train this network with backpropagation. And so the idea here is we know the labels we want. So we have the input. We say for this input, we want the output to look like this input. We just want it to reconstruct the input. So we're in some ways, it's kind of cheating to call it unsupervised learning because you are actually providing labels. You're saying, I want the input to look like itself. But we don't have to provide any external data. We just want to say, I want you to do this transformation such that this compressed version of the input happens to capture everything needed to create a decoded version, which basically is nearly identical to the input. That's the so-called autoencoder. And so what you end up with is the input and output of this bow tie are the same dimensionality, hopefully roughly the same signal. But the thing in the middle, Z, is a much smaller representation. And that compressed representation, hopefully, is sort of focused on the important discriminating features of the input and kind of throws out everything that isn't helpful to discriminate the inputs among each other. And that's kind of the idea there, is that then we can use that Z as a compressed version of X. And so we design the whole bow tie, but after training is done, we don't use the decoder anymore. It sort of is only there for training. This becomes the output right here, this Z. That's the idea behind the autoencoder. And you can view it as a nonlinear PCA. That's one way to kind of do it. But it's just a way to generate these compressed latent structures, representations of data that are useful for downstream processing, which I'll talk about next. So questions about this idea? And then just sort of a standard structure in basic neural networks, autoencoder. There are variations, like the variational autoencoder. There are variations of autoencoder used for different things. Very popular for things like anomaly detection. So, when you want to do anomaly detection, you've got a bunch of normal data, and it goes through, and you want to sort of be able to. So you say, what are the salient features of normal data? And then, well, this will produce kind of a model of normal data, and then if you get something that in the latent space doesn't have those features, then you're like, that's an anomaly. So that's an example of then you can, these are commonly used for things like cybersecurity anomaly detection. Actually, a lot of things these autoencoders are used for. Okay, so where do we see autoencoders in nature? All over the place. So this is an example. This also applies to mammalian olfaction. So you and me, but I'm going to just talk a little bit about insect olfaction here. So these are brains of 4 different insects. So this is Drosophila melanogaster, the so-called vinegar fly. So the kind of standard in genetics labs and so on. Red flower beetle, another common organism in laboratories, desert locust, and then the honeybee, which I'll focus on here. These insects, and actually most pan crustacea, so it turns out that insects and crustaceans, about, I don't know, 10 or 15 years ago, they were reclassified as being belonging to the same group. So insects, not arachnids, insects are like the crustaceans of the land, even though there are crustaceans on the land. And so crustaceans actually have a similar brain structure. Pancrustacea are kind of the older one, or sort of crustacea are kind of the older one. And so I'm going to focus here on this honeybee, but the point is, I hope you can see across these brains, just like across the brains of vertebrates, there's a lot of common structures. And so we're going to be focusing on these 2 in just a second here, but I just want to highlight there's this thing over here. This is called the optic lobe. It's so if you think about an insect, their eyes, this is inside the head of an insect, and their eyes are usually over here. And so those goes directly into this optic lobe. So that processes visual input. These are the antennal lobes. So if you think about an insect, they have 2 antennae that connect to their head right around where your nostrils are, and that's not an accident. Those are their nostrils, the antennae. Just imagine if you invert your nose so that you take all the stuff that's going on the inside of your nose and you put it on the outside of an antennae, and that's basically how they smell. And those are connected to one antenna goes to one, the other antenna goes to another. There are similar things that go on in your head with your nostrils. You may not realize it right now, but you're only breathing out of one nostril at a time. There's something called a nasal cycle. And so every 2 to 4 hours, it switches which nostril is you're breathing out of. This is why sometimes when you have a cold, it's like really annoying for a few hours and then it goes away, because you might have like one nostril that's clogged and the other that isn't. And so your brain can vasoconstrict parts of your nose to make it so that this nostril has stale or static air and this nostril is dynamic air and vice versa. And it just constantly is going back and forth. And there it appears like there are some odors that are better. There's some receptors that need dynamic air to fire, some receptors that need static air to fire. And so having one of your nostrils have static air and the other one have dynamic air actually helps you perceive odors. But old point I was getting here is you don't often think about your nostrils as like, it feels like it's just one big cavity, your nose, but it turns out there's 2 sensors being used in 2 different ways. And researchers, colleagues of mine down the hall from me, are actually studying how the antennae is being used asymmetrically in insects as well, as opposed to just 2 clubs that are out there. Because they'll do things like you puff odors at them and they'll do flagging behavior. Well, they're like one antennae will go over here and the other one will reach out. And so which one do they reach out for and so on. There seems to be a handedness in some insects where they prefer one antennae over the other and so on and so forth. Anyways, antennal lobes down there. And then your gray matter, where you do all your decision making, is there this thing called a mushroom body. And it looks like an inverted mushroom, or it kind of looks like a mushroom where the cap is going the wrong way. And it all of the sensory information from their eyes, from their noses, goes up their antennae, goes up into this mushroom body. And it turns out that you can do simple learning right in these organs. So you can learn basic colors, for example, right in these kind of more peripheral and organs. But then concepts like above, below, or whatever are learned up here. And there are ways that you can go into a brain like this, and with light, you can manipulate the gene expression in this brain so that certain neurons can be turned off when you flood the head with certain wavelengths of light. And you can turn off projections from these go back, undo whatever I did there. You can turn off projections from these systems up to the gray matter. And when you do that, they still can learn simple things, but they can't learn complex concepts. And so you really do have a compartmentalization of complex concepts up here, simple associations down here. So a lot of cool stuff in a tiny brain, relatively feed-forward structure, which also makes it nice to think about it when we're thinking about computational models. So anyway, I'm going to sort of just zoom this as like kind of a cartoon depiction of the honeybee brain. So a lot of this I've already kind of covered here. Antenna comes up, it connects to an antennal lobe. So what you're seeing here is the authors of that I borrowed this from. Everything on the left is actually on the right as well. Everything on the right is on the left. But to make it cleaner, they depicted some things on the left and some things on the right. So that's why this neuron here, this VUM neuron, is only shown here, but it actually also curls around this way too. But the idea is, so you had an antenna come up and it hits this antennal lobe, and it is each antennae is they're going to connect to these little bundle of grapes, which I'm gonna draw on the next slide, but it's kind of hard to see on this with this contrast. But there's little circles around the periphery of this depiction of the antennal lobe. And those are what are called glomeruli. Each one's called a glomerulus. And this is where our autoencoder lives. And so we'll get to that here in just a second. So, but the point is, antennal lobe comes in. This antennal lobe is effectively a feature extractor that generates a representation of complex odors that then gets projected up into higher order thinking in these mushroom bodies. And so those are those projection neurons that project information from this pre-processing step into the mushroom body. And just for fun, because we're talking about learning, and I talked about way back when we started this unit, that one of the ways you learn is you get neuromodulation, where your brain gets, when you get a reward, your brain gets flooded with a chemical that temporarily makes your synapses fire more or less readily, which will make it more likely for your short-term memories to get reinforced and then get turned into long-term memories later. And in the bee, that happens with this VUM neuron. And so you can touch sugar to even the antenna of a bee. And as that happens, this VUM neuron will flood a bunch of regions of the brain with this thing called octopamine, which is the invertebrate version of dopamine. And so gradually the brain gets washed over with dopamine, and all of those neurons that used to fire, when they got a sufficiently high stimulus, now get fire a little bit easier. So basically the brain's like, ooh, something just tasted good. Remember that. And it makes your brain, those synapses, much easier to fire. So it makes suddenly the brain ready to pick up, like hyperplasticity. It's able to pick up any associations it can possibly find that happened around the time of that reward. And that happens through this long neuron, which is basically just a delivery mechanism for dopamine all over the brain so that it can remember whatever the heck just happened that might have related to that reward. So it's kind of cool to see that simple reward mechanism there. All right, so that's our basic brain structure. So the next slide, I'm just going to focus on this antennal lobe and this mushroom body for my little autoencoder example. But any questions about this brain architecture here? Just basically the most invertebrates have a similar brain like this. Certainly the pancrustacea, definitely the insects, very similar. But the shapes and sizes of these things change, but the actual components are still there. Okay, and then if you're curious, I'll just point it out here. Notice that there's a projection neuron that goes to this thing that says LH, lateral horn. That's a part of the protocerebrum. So there's this this is a decoder for the brain. So basically you got all this cool stuff goes on in higher order learning, And then the brain needs to actually form an action. So the bee needs to like say, okay, that tasted good. I'm going to turn over there. So what causes the bee to turn? Well, there's a decoder which reads from the representations in the mushroom body that actually then forms the outputs, which then go out to the muscles. And that happens in this protocerebrum and this lateral horn. So that's what's going on here. So it's this, I mentioned in like reservoir machines, you've got this like these big central resources, and it turns out these projections that are going from the antennal lobe to the mushroom body, most of them are randomized. They're not trained, they're not plastic. So it's very much like a reservoir in a reservoir machine. And so then all that does gets trained is you get plasticity here in the pre-processing step, and you get plasticity here in this tiny little decoder. So you get this giant shared resource, which is basically just manipulating data, making it easier to process the data so that downstream you can use a really simple network. And that's very much like the reservoir machine, where it's like, I've got this shared resource that is not actually trainable, but it makes the world suddenly much more easy to process. And then I can build a really tiny network or a bunch of really tiny networks that can do complex things. Because now that I've pre-processed my data, I don't have to learn as much because the pre-processing step makes the data easier to understand. And that's basically what is happening here. You get this sort of easier to understand representation, and then these tiny little neuropills actually do the decision making for the insect. And it's similar for you. Okay. So autoencoders. So we got complex floral odors, very high dimensional. So you can take a puff of odor off a flower and you can do a mass spec basically on it. So you can look at all of the compounds that are in that flower, and there's going to be hundreds of compounds in that odor. And even if you have a flower of the same type, so you've got lilies, and all these lilies are next to each other. This lily's mixture of those compounds will be slightly different than this lily's mixture of those compounds, and so on and so on and so forth. So you might have been rewarded by a lily, but if your brain just uses a simple association circuit, then it will remember it was rewarded by that lily. It won't remember that it was rewarded by all of these other lilies too, because they're different enough that if you just do a simple discrimination, like you try to just do like an SLP, like if you just try to do some sort of separability or what, you're not going to be able to get to generalize and form a nice class of all the lilies. It'll just be like, oh, that reward is associated with this one flower, not this one type of flowers. So that's what's coming in to the nose, the antenna of the honeybee when it goes on a foraging task. And so that goes into, like I said, this antennal lobe. And this antennal lobe has all of these glomeruli. And so these glomeruli, there's, depending on the insect, there could be somewhere from tens to hundreds. There won't be thousands. There'll be fewer glomeruli here than there's dimensionality here. So these glomeruli, what I'm trying to get at is that they form a structure that allows for a lower dimensional representation of this input. And that's what happens. It turns out that every odor that gets puffed into here lights up an olfactory code. So you can actually do this thing called calcium dye imaging. You can make these glomeruli glow in a wavelength that is visible under a microscope, and whenever they turn on. And when you puff in a particular odor, then you'll see a code light up like I've hatched here. And you'll say, for this odor, this code lights up. And for this other odor, this code lights up, and so on. And as you start giving them odors that get clustered together, that are grouped together, like, oh, I just noticed that and rewards sometimes are involved here as well. So I'm kind of glossing over a lot of things here, but basically, as it starts learning that these flowers are important and other flowers aren't, then the representation of the lily starts converging. So it starts getting to that. You might have gone to the first lily and you might have gotten, all of these and also this one. The second lily didn't have this one, but had the rest of those. As it starts learning that lilies are important, then gradually every lily gets mapped to the same code. And every poppy gets mapped to a different code, and so on. And lilies might be in bloom this season and poppies aren't. So lilies are the ones you should do something with. And so these olfactory codes are what go into the mushroom body, which then lead to downstream decision making later. So what we see here is that this olfactory code here is a lower dimensional latent representation in a neural version of an autoencoder. And you have one of these as well, doing exactly the same thing in your head. So you go from high dimensional sensory experience to low dimensional representation for easier downstream processing. And that's what is going on here. So low dimensional representation for learning flowers or for learning anything. So autoencoders do exist in nature, this architecture. Questions about that? So basically, a binary, coding, ones and zeros, one for every glomerulus, one or zero for every glomerulus. And so there you've got a little bit code that you can code for each one of these odors. Okay. All right. So that was sort of like, that the message there was that we can transform the input with special circuits so that downstream clustering can be easier. We can do more than just organize the input. We can actually actively try to predict it and use those prediction structures to better facilitate learning as well. And this is so-called predictive coding. It's active structure generation in the brain for minimizing surprise. The sort of idea here is that you've got a prediction stream where your top-down centers of your brain, coarser grain centers of your brain, make guesses about incoming sensory data. So what they think sensory data will look like at the lower level. So then from those higher brain regions, you're predicting what the low dimension, what the lower level, but higher dimensional representations are going to look like. And then from there, you're going to then compare those to what you actually see or hear or smell, and then use that error to update your model for that prediction. And so I'll give an example of this in a second here, but that's the idea. It's almost like back propagation. You've got a high level, you predict what the high dimensional input is going to look like next. You get a high dimensional input, you look at the error, and then you update your model at the high level to make better predictions later. And so one example of this comes from reafference and efference copy. And so I'll just sort of maybe draw this diagram out. So when I reach for something, I send a motor command out to my muscles. That is what's called an efference. It goes out from my brain to my motor system. I then receive input from my moving arm, and that's called a reafference. And so it is sensory input generated by me being collected by my body. Now, my brain actually has a model, and you can find these models inside brains, where it gets a copy of the motor signal. And that model predicts what the reafference should look like if everything worked out correctly. And when my reafferent signal coming in doesn't match what my model of my muscles should do was, then I get a discrepancy, which my brain uses, in this case, not to update the model, but it actually uses it to say, oh, you meant to move your arm here, but you hit something. So that was an X-afference. I got other sensory disturbance. You need to push a little harder in order to try to get things where you were going, to deal with this disturbance. But alternatively, and what your other systems do, you can also use that discrepancy to fix the model. And gradually over time, fixing the model, that will help you better understand the world around you. And this is what happens in vision, in audition. So when I talk, I'm not surprised to hear my voice, but I guarantee you a baby is surprised to hear the first cries it hears. When my eyes saccade around the room, I don't notice it. But a baby, it its world is freaking out. Um, and it's because it hasn't built up models that predict what's going to happen next. And so it can't filter out these things. So you have stable vision because your brain is able to predict, like when it moves your eye around, it knows inside its model, oh, I'm about to move the eye to the left. So the vision is going to move to the left. So your brain accommodates for that. So you don't even notice it. But if you look at the signals come out of your eyes, they're freaking out. They're shaking up all over the place. But your brain filters that out. And that's because of this predictive coding. Likewise, with audition, you ignore your own voice. In this particular case, you can't tickle yourself. That because you are predicting what's happening, and so that you don't have a discrepancy when you try to tickle yourself, and so the tickle doesn't come through. The reason I'm bringing all this up and I'm rushing through this a little bit here is I just want to make this connection is that this is the biological version of what we refer to in machine learning as self-supervised learning. So the idea here is that predictive coding minimizes surprise. Your babies move the limbs, they build up models. Rats, I've mentioned this example before. Rats that were allowed into a maze before there were any food rewards later when you give them food rewards can go straight to the reward even though they weren't ever rewarded that beforehand. Rats that are naive to the maze, even though there's a food reward, now have to search throughout all the maze and it takes them a long time to find the food. So there's this so-called latent learning where your brain is just constantly updating predictive models. And that is what we refer to as self-supervised learning. And I wanted to make sure we at least got there because this is huge. GPT is all self-supervised learning. There's a little bit of fine tuning at the end, but the idea behind BERT, which is sort of behind it's an earlier model for how Google did things like sentiment analysis and so on, and GPT, basically what these models do is instead of giving them supervised targets to follow, they say, here's a big data set. You why don't you play with the data set and then try to predict the manipulations that you've made? So there's an iterative process inside GPT, but when it's being pre-trained, where you give it a bunch of data and it truncates. In the case of GPT, this is different for BERT, but it cuts off. Like if you have a bunch of data, like the quick brown fox, the text was the quick brown fox jumped over the lazy whatever, et cetera, et cetera. It has a supervisor that is built into it that will take this data and will cut it off here and then ask the model to predict the next word. And it will generate its own training set by doing that over and over and over and over again, and then uses just back propagation, just like the autoencoder, to then train itself. And then that is then what allows GPT to then respond to your queries later. It does not, there is some fine tuning that's added onto it, which is a little bit more supervised, but most of it happens in this self-supervised layer. That's why it's so-called pre-trained. That's what they're referring to there, is that it is a tool which just played with an input data set in order to figure out patterns so that later when you ask it, when you give it your own prompts, it then fulfills the patterns. And that's just self-supervised learning. And so, don't really have time for the rest of this here, but basically extending the idea that BERT was a tool that Google used, several years ago to start, understanding web pages, what was on those web pages, works in a very similar way. So that's basically my takeaway. This is just sort of a summary of everything we've talked about already. Next time, I don't want to keep you any longer, and I'm sorry I was a couple minutes late. We'll, finally connect to the neuroplasticity to say, okay, how does this underlying fundamental neuroplasticity work? And that's what we'll talk about spiking and neuromorphics. So with that, that's all I got for you. So we'll see you Thursday. Uh-huh.